Bit by bit, the human genome is giving up its secrets. If we can find out what our genes do, then we could put that information to work, to create the perfect child.
A man and a woman that want to have a child will walk into a fertility clinic, and the doctors will take 100 to 200 eggs out of the woman. They'll be fertilized with the man's sperm, and then they'll do a genetic profile on all 200 embryos and choose the embryo that has the combination of genes from the two parents that the parents want to see in their child. That's where they'll start.
But as we learn more about our genes, there's a chance that some parents will go beyond choosing embryos for health and select them for more trivial reasons.
I have no doubt that in the future, people are going to want to use this technology for uses that go beyond medicine, for uses that are cosmetic, for things like eye color and height, and other things that are on the border of medicine like longevity. I have no question in my mind this technology is going to be used to provide people with these kinds of genetic choices in their children.
Even if some parents and their doctors were willing to use PGD to make designer children, the technique is limited in a crucial way. PGD can only select an embryo with genes inherited from the parents.
None of these embryos have genes the parents themselves don't have. And so if the parents want their children to be very tall or to have a particular talent that's not present in their heritage, then they're not going to be able to get it just by choosing among their embryos, but they will be able to get it by adding genes into the embryos.
One day, parents may be able to pick any gene they desire from a range of bottled genes and have it put into their embryos. It's strictly against the law to do this in humans, and yet scientists have been quietly perfecting the art of adding genes into embryos for years. In animals.
The very first gene that was added to an embryo was a gene coding for growth hormone. It's a gene that causes an animal to grow larger. And a growth hormone gene from a rat was placed into a mouse embryo. And when the mouse was born and it grew up, it grew to twice the size of a normal mouse. This had a horrifying, incredible effect on society at large. I mean, this was the first instance of genetic engineering actually occurring in an animal.
This is how they made the super mice. Thousands of growth hormone genes from rats were injected directly into fertilized mouse eggs. As the eggs divided, this new gene was copied into every single cell of the developing pup, including its egg or sperm cells, the so-called germ cells. The offspring of these mice would also be giants, their genetic blueprint changed forever. But scientists knew genes could never be put into human embryos this way. The risks are far too great.
The process is very inefficient. And so perhaps 1 out of 100 mice will be born with the appropriate genetic engineering occurring. And so I can breed 100 mice, take the one mouse out of 100 that has the correct gene change, and get rid of the other 99 mice. You can't do that with human babies. And so as long as the efficiency was so poor, it wasn't conceivable to do this with humans.
Compared to normal chromosomes seen here in blue, the artificial chromosome was tiny. A speck of pink amongst the blue giants. So far, it's only been designed to carry a single gene, but in principle, an artificial chromosome carrying any number of genes could be built.
With the creation of an artificial chromosome in the laboratory, we can create a brand new chromosome which has hundreds of new genes, and then we can take that entire chromosome, put it back into the embryo whole, and it gives us much, much greater capacity to modify the genome.
One day, it might even be possible to buy artificial chromosomes off the shelf. A chromosome with all the genes for perfect physique, another one designed for long life. Some even think a chromosome for added intelligence could be possible.
DNA. How does it function? DNA is extremely complex. This model shows only part of the long chain of the many thousands of atoms comprising a given DNA molecule. Here's a simplified, diagrammatic version of part of the DNA chain. It still looks pretty complicated, so let's see how it's built up. There are two main strands, wound round each other in the form of a double helix. Each strand is made up of phosphate groups, here coloured yellow, and deoxyribose groups, coloured green. The two strands are linked together by basic groups which are attached to the deoxyriboses. The bases on one strand are joined to those on the other by hydrogen bonds. 4 different bases are involved: cytosine, which can only link with guanine, and thymine, which can only link with adenine. The bases can only pair in this way: cytosine with guanine and thymine with adenine. You can see that here. C always linked with G, A with T. So, DNA consists of long-chain molecules in which 2 spiraling strands are linked by pairs of bases in the way we've just shown. The bases may occur in different orders in different DNA molecules. Now, an important feature of DNA is that it can replicate itself, make copies of itself. This is how it does it. The 2 strands come apart. Each separate strand now builds up a new strand alongside. The new strand is built up from separate nucleotides, each a combination of a phosphate group and a deoxyribose group to which one of the 4 bases is attached. The base finds its complementary base on the DNA strand. Here the base is thymine, which links up with adenine. Here, cytosine links up with guanine. And so on, until a DNA double helix has been formed once more. The same thing happens to the other strand from the original DNA molecule, so that now we have 2 DNA molecules. This is how a given DNA molecule can make any number of copies of itself. How does DNA carry genetic information so that a particular plant or animal develops in the way it does? Slightly simplified, it works like this. The spiral unzips again, and this time a slightly different strand builds up alongside the DNA strand. This is ribonucleic acid, RNA, in a form called messenger RNA. It's like DNA, except that the green deoxyribose groups are replaced by ribose groups, here coloured orange, and at the same time the base thymine present in DNA is replaced by uracil, here coloured purple. The sequence of bases along the RNA strand matches the sequence of bases along the DNA. This messenger RNA, and remember this is only a short portion of a very long chain, now moves out from the cell nucleus where the DNA is to the ribosomes in the cell. Here, proteins are built up. This is how the process works. Molecules of transfer RNA, each with groups of 3 of the 4 bases, attach themselves. Each carries a particular amino acid, depending on the sequence of the 3 bases. This one with uracil, adenine and cytosine carries an amino acid represented by the grey rectangle. It finds its place by base pairing on the chain of messenger RNA. A chain of amino acids is built up, each amino acid corresponding to a particular triplet of bases or anticodon on the transfer RNA, which fit the corresponding triplet or codon on the messenger RNA.